I am Vinita.
Hello, I'm Shuman. So today we have with us Joaquín Navas Acedo, who is born in Spain, where he completed his undergraduate and master in genetics and cellular biology from Universidad Autónoma de Madrid. Following his masters, he moved to Marine Biology Laboratory at Woods Hole, Massachusetts, United States of America, where he did a fascinating course on embryology, concepts and techniques in modern development biology.
Nawaz did his postdoctoral research from Dr. Jana's lab at Stover's Institute of Medical Research. He went back to MBL at Woodhull, Massachusetts, for a teaching assistantship in the same course he took while he was a student. He is currently pursuing his postdoc in Dr. Allender's lab in Biozentrum of University of Basel, Switzerland. We would request you to kindly present your work.
Hello. Thank you very much for the invite and for the introduction. It is really exciting being here today. And I think that the job you're doing is fantastic and everybody should watch it. The videos are fantastic and super fun to watch. So my journey through science goes from the middle of Spain to the middle of the US to the middle of Europe. So during my bachelor's, my undergrad, I went to Fernando de Castro's laboratory. It was in Toledo, which is, if you take a bus from Madrid, it's like a 40-minute south of Madrid. It's a beautiful city. And there's a hospital there for paraplegic people. And then they do basic research. So I decided to join his laboratory because he was doing neurodevelopmental biology. And he studies the olfactory system of mice and how the olfactory sensory neurons project to the olfactory bulb in the brain, and like how this affects the behavior. So when I joined, I had no laboratory experience. So everything was new to me. Like doing a PCR or a Western blot or an immuno, all these cell culture, everything was fun, right? And I had the time of my life and I learned a lot. So then I went to the Instituto Cajal, or the Cajal Institute in Madrid, to the lab of Juanjo Garrido. There I pursued what I think is one of the most fun things you can do, which is to study how neurons go from looking like a ball to looking like a neuron, right? With all the dendrites and the axon, how they grow. So I studied this signaling pathway called the purinergic signaling, which is mediated by ATP, ADP, AMP, etc., and how this controls the axonal cytoskeleton of hippocampal neurons. And it was awesome. I learned a lot. Became an expert on Western blots, and I'm now forgotten a lot of that, but I did a lot of Western blots, and it was a really fun experience. A very small laboratory. It was him and me, and then that's it, because everybody else like either graduated or just move on. And then when I was at the Cajal Institute, as I said, I wanted to go and do a PhD, and then I considered both Europe and the US. And then I applied to several places in the US because the deadline was sooner than in Europe. Like I think the deadline in the US is mostly like December, and then in Europe is like latest late January. So they invited me first to go to the US before I ended up applying for Europe. So and then I joined the Stowers Institute, which is a very particular place. So they have the money and the resources for you to basically do whatever you want. It's a very tight community, and they have a very strong core facility network within the institute. So you can do like imaging, mass spectrometry, molecular biology, whatever you want. So I joined the grad school, and then I did rotations in very fun laboratories. So I rotated with Alejandro Sanchez Alvarado, which works in planarians, these magicians of regeneration, and that was fun, but it was not for me. And then I rotated with Paul Trainer, who was in the development of the craniofacial structures and nuclear cells in mouse and zebrafish, but also in fun critters like lizards and chameleons, and all of these very cool stuff. And that was fun, but it wasn't for me. And then I rotated with Tatiana, and then I just fell in love with what they do, because and I'm going to show you why. So Tatiana studies the lateral line of zebrafish, which is a model for morphogenesis and regeneration, because so the lateral line of fish develops from what you see in the screen, which is called the lateral line primordium, which is this group of cells that undergoes coordinated migration. And then on its way, it deposits this cluster of cells called the neuromast. And Tatiana's work and other people's work showed the signaling molecules that let this group of cells migrate coordinated. So it's chemokines and Wnt signaling, FGF signaling. The first time I saw this video, I was like, that's it for me. I want to join this laboratory. But what I studied was how these group of cells called the neuromast detect water movement, because all fish have this lateral line, which is like this group of cells are all over the body, and they detect the water movement coming from any direction, right? And they do this because within this clump of cells, there is a very specialized cell called the Herschel, which has what is called a cilium or a kinesium, which gets deflected with the water movement. And these cells, actually, we got them in the inner ear, and the ones that let us hear, and are the ones that detect when we do this with the head, or when you're going too fast in the car. And what is fascinating about zebrafish is that when we lose these cells, we go deaf. Like you get very sick, and you get a shot of antibiotics, you lose these hair cells, or you undergo chemotherapy, you lose it, or as you get old, you start losing this hearing. But what zebrafish can do is to generate more of this, right? So they lose them, they don't care. Like throughout their life, they keep producing them. So what I studied was in this, which was very fascinating to me. But what I studied was these cells need to be arranged in a very specific way, in a planar polarized way. So they all arranged like in a coordinated pattern, and for the animal to properly detect the water movement. And what my studies during my PhD was to study signaling pathways that control this organized arrangement. And then with this, we discover new mutants and new processes during this coordinated Herschel orientation. And that was very fun. I got to do a lot of immunos, a lot of imaging, and take a lot of videos and look at zebrafish, and let this go for a very long time, which I absolutely loved. And it was a fantastic experience. It completely changed the way I see science. And then for my postdoc in Switzerland, I joined Alex's lab. And then now I study sensory neurons, but of a different kind. So these sensory neuron that you see is called a Rohenbeer neuron, which are neurons that live in the dorsal part of the spinal cord and then send these beautiful projections to the periphery. And they innervate the skin and the muscle, and then basically used to detect touch or temperature or obnoxious stimuli. And the question that I'm pursuing now during my postdoc is this fascinating process in fish and frogs. So it doesn't happen in other vertebrates. So these Rohenbeer neurons early during development form the primary somatosensory system. And then these die, and they get replaced by a second set of somatosensory neurons called the neurons of the DRG, or the dorsal root ganglion. So what I'm studying is how you replace this primary somatosensory system by the secondary somatosensory system, and how these two systems are similar to each other or different from each other. And, the kind of neurotransmitters they express, how do you produce from a progenitor, how do you produce a Brohan-Beer neuron or a neuron at the DRG? And what are the dependencies between the, one and another? Because I think we can use this to understand how the nervous system matures, because there are other examples in many other vertebrates, are not zebrafish and frogs, but also in invertebrates of so-called pioneer neurons that get there first and then maybe shape the behavior of the neurons that come after them. And this has been so fun so far. I get to do still, a lot of imaging, but then now I'm, learning how to do transcriptomics at the single cell level. I'm learning how to program and to code in R. And as I said, it's, this book gets summarized by my interest in developmental biology has been always, how do you go from, how do cells know what to do? And, how do you go from, like one cell, you fertilize it, and then they somehow know how to make all these things, right? And all animals have found their own way of doing this, right? Like we know from developmental biology that, some animals gastrulate in a specific way, and then they some other animals gastrulate in a different way. Some animals more, some animals grow less. Some animals, use some molecules. Some animals use the same molecule for something else, right? And then it just works, right?
Your enthusiasm is really infectious. It's fascinating, honestly. I got goosebumps seeing those videos. Like, it was that amazing. And I think, I mean, more than a scientist, I feel that you are also an artist. Like, these pictures are so cool.
Yeah.
I like drawing a lot. I'm actually, I have the kind of abstract vision that I need to draw. Like every time I want to do an experiment, or I have a result or something, I need to draw it, because otherwise I don't see it. Like if I want to do a cloning, right, or I want to do like whatever, if I don't see it, I don't understand.
So, do you have any your life incident to share from where you fell in love with science, or from where you understood that this is the thing you would do for rest of your life?
My dad had a microscope, and he will take like the classical story. You take a drop of rain and then you look at it, and you see all these things happening. And I thought that was pretty cool. But I had just an interest in science. I didn't know that what science was. I just liked to know things. And then I used to read a lot of like books about like medicine or biology or history or sociology, because they were full of data, right? So I just wanted to learn, how, when something happened or how something happened, right?
Discovering different facets of ourselves is one of the most fascinating things, right? And that brings meaning to our existence. So, what was the best thing you discovered about yourself?
One thing I always knew, but then now that this is my job, it is more clear to me, is that I cannot force myself to memorize things. Like never in my life, studying, sitting down and then, trying to memorize things. It's either it clicked for me or it didn't. And the other thing that I learned about myself is that if you know how to approach it, it is okay being a mess sometimes. Because my mom, it drove her crazy, what a mess I was. But then within that mess, there was some kind of order. And then throughout the years, I learned how to control that mess. And then, if you can control that, I think it's okay. And it's, you have to allow yourself some space for messiness and chaos, because sometimes sometimes it's okay.
So, what do you think that how you as a person have changed in all these years?
Well, I changed a lot. I have to say that, I was very lucky to go to the US, and then that automatically made me very privileged, and also automatically made me an immigrant. And then, that changes how you see the world and how the world sees you. Because the moment you leave, your country, you're the one with the accent. And then that's one thing that you learn about yourself, that, if you're not in your home country, you have an accent, and that's okay. But also, like how privileged I was, like myself, I'm, able-bodied white guy, and that makes me the majority in the world, right? And also made me realize that I could, or I had to, that was a morally thing to do, use my privilege to make science fun for everybody. So I tend to educate myself, and I try to educate myself on how to use this that I have to make science accessible for everybody. And then another thing I learned is how much I had to work on myself. So when I joined the laboratory, I have a broad interest on many things. That's what Tatiana used to say. And that made me do experiments, 12 experiments at a time about like 12 topics that were different from each other. And then you realize that that's okay, but that's very unproductive. And then you learn how to control that, and then develop a drive and a focused mentality on, it's okay having a broad interest, but then you need to have priorities on, when to do something, how to do something, and develop a strict work ethic. Because otherwise you end up doing nothing, right? Or half-assing everything. So it is okay to, be interested in many things. And I encourage everybody to do, to have secret projects, and you don't tell about your PI. But then at some point you need to own it, and then like push it forward so you get somewhere, because otherwise, like it's you could use your time doing something else.
So what are your, like, how do you deal with failures? What are your combat mechanism when you fail?
So failures, like, let's be straight, failures are tough. And whoever tells you that, it is not, they're fooling you. They're trying to sell you something. Failures are tough. What I usually do is, okay, I fail, and then you own it. And then what I do is to sit down and then think about what happened, right? What did I do, right? How did I do it? And then what is the thing that I could change to figure out what went wrong, right? Sometimes, you can just repeat it right away, like you do a PCR and then you run it, and then there's no bands. And then it's like, okay, I have no bands. And then there's, like a number of things that you can change, and then you can just repeat it right away and then see if it works. But sometimes you spend a lot of time doing an experiment, and then it doesn't work. And then in that case, what I do is to sit down, think about it, and then I leave it for a day, and then come back with fresh eyes, and in the meantime, you do something else, and then you come back, you look at it with fresh eyes. And then, I think about what I can do, and, like the plan A, plan B, plan C, plan D, etc. And then hammer it down until I can say I solved it, or I did everything that I could to solve it, and then I have no idea what to do. The other thing that helps a lot is to talk to people, because you cannot pretend knowing everything. And I only know a very few people that know about pretty much everything, but I'm not expert in everything. And then I can go and ask my neighbor in the laboratory, or my neighbor in the floor, or even somebody on the internet, that all these connections that you make. Hey, do you have time to chat? And then you explain it, and then they say, oh yeah, when I was a postdoc or a student, I tried to do something like that, and it turns out that this enzyme does not like magnesium chloride, and then boom. Or maybe sometimes people do not have an idea of what how to solve it, and then you just have to be creative and hammer it down enough that it gets solved, or it doesn't. Sometimes it doesn't work. So if you fail at an experiment, that's okay. It doesn't mean that you have failed as a scientist or anything.
Your experimental images have been featured in such popular magazine covers such as Zebrafish, The Scientist, Developmental Dynamics, and they are so insanely beautiful. So, what is your secret to perfect an experiment and make it into a canvas, like a masterpiece?
So I think that the secret to perfect an experiment, I think, is two things. You need experience, right? So you need to first learn how to do the experiment. So somebody had to teach you, or you had to learn how to do it yourself. And you need to do it a number of times, and then it fails, and then you troubleshoot every single step, right? But also you need to learn what everything does in the protocol, right? Like, what does the magnesium chloride do in a PCR, right? Like, why do we put magnesium chloride, or why do we put DMSO sometimes? I think that the second thing you need to perfect an experiment is, as I said, you need to surround you with smart people. And you need to build this network of people that you trust, and then you value, and then people that know, with different expertise, right? Molecular biologists, developmental biologists, cell biologists, geneticists, zebrafish, Drosophila, C. elegans, like whatever, right? I think that it is very important because it gives you perspective. Yeah, having a good network of people you trust and value is very, very important, because science does not happen in a vacuum.
Though COVID-19 has been quite tragic and has created quite a havoc globally, it has also taught us quite a lot of important lessons, and we have discovered new ways of conducting day to day activities like webinars. So, do you think COVID-19 has brought the science community closer through social media?
So I think that, in a way, um, COVID-19 has brought the science community closer, in a way, meaning that, like you very well said, that now we do, the Zoom seminars, or you attend conferences virtually, which, was a new thing. And everybody, I think that now everybody can participate wherever they are, right? But on the other hand, I think that some people have seen it maybe as a minor inconvenience, or they have not been affected as much by the pandemic. And there's data, showing that the pandemic has impacted women, for instance, more. So I think that unless people, or we as a community, realize that doesn't matter how many virtual conferences we do, that it's just like an impact that affects only a group of people. I don't think that's fair, right? I think that until the community does not come together to solve that problem, then I don't think we can say that the community is closer, right? I mean, community is closer for some things, but not for everything, right? So until it's not inclusive for everybody, then it's not inclusive, right?
Recommendation letters are a very important part of the application process, and in most reputed universities, and it has both its pros and cons. So what is your opinion on it?
I think that I was very lucky and very fortunate to have undergrad mentors that wrote letters of recommendation for me. And for that, I will always be grateful. And the same for when I apply for postdocs. So I will also say that letters of recommendation and, like making it in science is not only who you are, but who you know. The only thing we can strive for, I think, is having more, making science more inclusive, so there is more minorities in positions of power that can write good letters of recommendation, and then promote more underrepresented minorities to, like, to basically first put them in the pipeline and then keep them in the pipeline, right? Without that, it's just, you know, white dudes writing recommendation letters, right? But I don't see that happening anytime soon. I mean, there's been some progress about it, but I think that we still need to work on that. And then I don't have any suggestion to replace recommendation letters. I think that if there was a better way of doing it, I guess somebody will have already proposed it. Maybe, you know, an interview is a thing that it's how people talk about you, but also people like that activation energy of putting your foot inside, right? Like if you can just get your foot inside and then show who you are, then I think it's just it will be much better, right? Because the majority of people do not get that opportunity to show who they are, right? Like it's just a letter from somebody saying this is, you know, these are the qualities, and then it's up to the person to decide, okay, do I like this or I don't? And maybe with like a plain interview, it will be more clear, or it will be an additional layer to add, and, you know, like to make it more clear of who you are and what you can do.
When we get into science, we hardly ever consider the possibility of losing interest in doing science. It is very natural and human to have interest shifted to something else, like you said earlier. However, the thought of facing judgments can be overwhelming. So, what would you do if you ever fell out of science?
I do not know. So far, I love science. Maybe I will be like a comic book artist or something like that, because I like drawing very much, and I have friends that, you know, draw and color for comic books. But I guess I will do something else, right? I mean, science is just a job, right? You go to grad school, you get your PhD, and then you don't necessarily need to pursue a career in academia, right? You can go and, you know, because you develop some skills that are useful somewhere else, right? So I guess I will try to figure out, you know, what I like, and then just find another job, right?
Recently, I have been reading few science comic books, and those are quite fascinating. I even went through a few poems and like science poems, and I feel that it is very cool to communicate science in such a way so that commoners can also understand it, because I guess ultimately they are contributing through their taxes towards the science that we are doing. And I think science is for all, like everybody is doing it for everybody.
Yes.
So yeah, so that way I think they also have the right to know what is going on.
Superhero movies are thrilling because they make us imagine all that we could have done if we had those powers, right? Uh, if you were given the power to change something, what would be that one thing that you would want to change about academia the most?
As I said, inclusivity. So I think that it is the moral thing to do, make it accessible for everybody, because it's not only the moral thing to do, but also it just makes it more fun, right? If everybody can participate, which implies, you know, a number of things, but I think that one of the things it implies is getting rid of bullies. So people that abuse their power, and also getting people in the first place. So, you know, like, you know, science communication, reach out to people that otherwise wouldn't hear about, like, what, you know, what we scientists do and how we do it, and, you know, like in communities that wouldn't have access to that, giving them the opportunity to get their foot in, but also to keep that people inside once they're inside. So the fixing the leaky pipeline.
So I have always imagined books as windows to a different world. Is there a particular book that is a must read according to you?
Ooh. So non-science, I will recommend everything by Terry Pratchett. Like the This World series is amazing, and I recommend everybody reading it. The same as Douglas Adams. It's just that humor that they had and how they talked about just the world and the problems of the world in a way that, you know, it was humorous, but also it was a criticism to the world, right? And then as for, you know, for developmental biologists, I would recommend Rita Levi-Montalcini's biography, because it's fascinating. That woman was badass. Like, yeah, I mean, it's just from, you know, from her, you know, childhood all the way to, you know, getting to be a scientist, when the world didn't want her to be a scientist, and then how she pursued her, you know, through hard work, but also because, you know, she was stubborn enough to say, I want to be a scientist and I want to study this. And then, you know, when she moved to the US, and then the, you know, the whole neurotrophic theory and the beautiful experiments she did, and I think it's just fantastic.
Do you think that breaks help you a lot to rethink about the science you're doing and the questions that you want to address?
Absolutely, yeah, absolutely. I am guilty of working too many days and too many hours. But I absolutely believe that taking a break is something that is important, and everybody should do it. Yeah, if you are burned out, you're not help. You are of no help to anybody, not even to yourself.
So, what is your mantra of work-life balance?
I think that one should work as much as they want. Um, so, you know, one day you feel like, you know, you work for 4 hours and you're done, then I think that's it. Because if you want to stay for another hour, 4 hours, just not doing work, or, you know, like not doing anything, then, you know, you better be at home. If you had to put 11 hours one day because it's, you know, like experiment that requires you to, you know, to stay that many hours in the laboratory, I think that's okay too. As long as you're driven and you're focused, then you can just make it happen, right?
Is there any mistake that you have done in your journey and would advise young students to be careful of?
Made like anybody else, you know, too many mistakes, right? It's just part of life. I think that it is important that when you make a mistake, you learn from it, and then you can, you know, you can fix it. I will advise, like, if I could give one advice to, you know, people that are watching this video and, you know, want to go into science, you know, like any kind of science, I will say, don't sell yourself too short and don't spread yourself too thin, because I think that you are smarter than you think, and you work harder than you think. So then own it, and then you know, if you had tell people, just tell them, right? Because you put all those hours in, and you know, it's just own it, right? I mean, you've done it, you've done the good job, right? So just do it, and then don't spread yourself too thin. It is good having many interests, but, you know, you need to develop a sense of urgency, like my boss says, right? You need to, from all the things you could do, you need to know what to do, and then, you know, like to prioritize, and then just go and do it, because otherwise you're not gonna get things done. So yeah, that will be it. And then you know, like I said, you are smarter than you think. Don't let anybody tell you otherwise, or let other people decide how smart you are. You know how smart you are.
In your whole career of PhD and postdoc, you must have mentored a lot of young students, like young undergrads. So, what do you think is a mentoring advice, or like what do you think that a mentor must have?
So there are many styles of mentoring. So I can only speak about my style of mentoring, which is I try to listen, and if I know something, you know, if I can teach somebody how to do it, I will do it. And I can, you know, I can show you as many times as you want. And then I think that one needs to be kind, right? Because everybody's circumstances are different, and you don't know what the other person's circumstances are. So just try to be kind, and then to understand. Just be there, right? I think that being there is one thing. The cheapest, most powerful thing that you can do is just being there. And then from your mentors, try to pick what you think is the best qualities of them, right? Like if you like something that one of your mentors does, then do it yourself, right? I mean, why not?
Even I feel that it is very important to collect the good things from mentors and apply it in you. Like not everything in one mentor, not everything will be absolutely perfect. And so yeah, we can just collect the good things from everybody. So now we are coming to an end of this interview, and the last question I would like to ask you is that what do you feel about Kitchen Lab?
I think it's awesome. Like I had no idea this existed, and then you told me about it, and then I think it's great. Like I think what you are doing is fantastic, and I think it's great. Like I watched the other interviews, and I think it's fascinating just listening to other people's journey, and then encouraging people to pursue, if they want a career in science. It's very important, because some people do not know that they can pursue a career in science, because they never had their first generation, they never had anybody from their families going to the university, or to have higher education, or they don't know anybody from their family, or like the extended family, that they do science, so they don't know that's an option, right? I think that is okay, and it's a moral duty to tell everybody that they are welcome in science, that then they should do science if that's what they like. And then once they make it, if they make it, to help other people realize that they can do science, right? That's why I have the utmost respect for people that do science communication, because it's very, very hard to do science communication, and very, very hard to find a good style to let people know, that it's okay to do science and to be excited about things. So I really appreciate what you do here in Kitchen Lab, because I think it's a very good job, and I think it's a very important job, and I hope that you get a lot more people to participate in this. I'm gonna tell everybody to participate in this.
Thank you so much.